My name is Robin Hall and I work at the Ontario Institute for Cancer Research. This is my abbreviated poster presentation about the Reactome project. Reactome is an open source, open access, expert authored, manually curated and peer-reviewed pathway knowledge base. We've developed intuitive software tools for data visualization, integration and analysis. The central component of our pathway curation is our data model, which is based upon the reaction that allows us to capture many different types, including drug-target interactions. Typically, the drugs we curate bind their target, and the resultant complex regulates the target reaction. Reactions are then grouped together to form pathways, and pathways can be grouped into human recognizable biological events, including altered pathways corresponding to human disease. To date, we've curated over 2,000 variant proteins and their involvement in disease pathways. The pathway browser is the main visualization tool to browse biological pathways. In this pathway browser widget, the pathway hierarchy is in the left panel, the diagram is in the right, and a details panel below the diagram provides information as the user interacts with the pathway diagram. The pathway browser provides a framework for data analysis and visualization of external datasets. As you drill down from the pathway overview summary into specific pathways, you can see the specific textbook illustrations or pathway diagrams with experimental data overlays. New to Reactome is the high-level pathway overview visualization based on Voronoi tessellation. To support multi-omics data analysis, we have also created a new Reactome GSA service that uses our existing web interface and a novel R bioconductor package. Reactome GSA supports the direct importation of data from the Expression Atlas, the single-cell Expression Atlas, as well as user-supplied data, and offers different analysis methods. The Reactome FI Viz app performs single and multi-omics pathway and network-based data analysis via the Cytoscape platform. Following a recent update, the FI network now consists of 13,000 proteins and 436,000 interactions and provides support for gene set enrichment analysis for Reactome pathways. To support data scientists, we've developed a Python package by wrapping our APIs, including the analysis and content services and the Reactome FI Viz app. We've also developed the documentation and deployed the packages into several Python repositories and provided a Docker file. Now I'd like to talk about a project that we're working on with the Illuminating the Druggable Genome program, where we're developing a portal by integrating IDG data to provide a pathway-centric view for understudied human proteins. The IDG protein classification scheme, called Target Development Levels, categorizes proteins into 4 categories: T-clin, T-chem, T-bio, and T-dark. We can see the 4 IDG development levels associated with Reactome entities for the G-alpha signaling pathway. The color overlay is as follows: dark blue for the T-clin level, light blue for T-chem, orange for T-bio, and red for T-dark. Zooming into the diagram to view the olfactory receptor entity, we see these T-dark proteins are. In the past, we would have had to have performed pathway enrichment analysis to overlay this data onto the pathway diagrams. The portal also supports the overlay of expression data collected from the IDG project, for example, human proteome map protein expression data for heart and liver. The focus is on one reaction, and on average, proteins involved in this reaction have higher expression in the heart than in the liver. Users can toggle to a simplified FI view, which shows clearly that RYR2 and RYR3 and several other proteins have higher protein expression in the heart than in the liver. We've also implemented features to overlay pairwise relationships together with target development level information. And in this table on the left, we can also see the drugs that are known to interact with the members of the RYI tetramer complex. Here's a summary of my talk. I'd like to take a moment to acknowledge the contributions of the Reactome Consortium members and support from the funding agencies. Thank you, and I'd be happy to take questions.